Clicker questions for Chapter 7, Transcriptional Regulation. 1. Why is the lac operon under transcriptional regulation? A. Transcriptional regulation allows the cell to economize by not investing energy in the synthesis of unnecessary mRNAs and proteins. B. Lactose is the preferred, but not the only, beta-galactoside that is biodegraded by beta-galactosidase. If the lac operon were not under some form of control, all the beta-galactosides in the cell would be degraded. Letter C. In the human gut, E. coli cells rarely see lactose, a disaccharide found in milk. It would not be able to make sense to synthesize a series of enzymes to degrade a substance that is rarely encountered. Or D. The presence of too much lactose could be toxic to E. coli cells, so there must be a mechanism to prevent its buildup. Answer is A. Transcriptional regulation allows the cell to economize by not investing energy in the synthesis of unnecessary mRNAs and proteins. So very simply, if there is glucose present, then there's no need to have anything that will break down lactose, as it is more beneficial, more efficient for the E. coli cells to break down the glucose as long as it is there. Number 2, discuss a potential answer to the lactose paradox. If lactose needed to induce the synthesis of the lactose permease, which is needed for the cell to take up lactose, how does lactose get into the E. coli cell to activate the lac operon? Letter A, lactose can freely pass across the cell membrane. The permease is needed only to speed up the rate of the uptake. Or B, lactose can freely pass across the cell membrane. The permease gene is the vestigial remnant that serves no purpose, and that natural selection has yet to remove. C, the lac repressor occasionally falls off or debinds, which allows a minimal level of 3 enzymes needed to take up and metabolize lactose. Or D, occasionally, transcription and translation of the lac repressor gene generates a defective lac repressor protein. This defective protein cannot repress the expression of the lac operon, so a certain number of permease proteins are always present. The most likely answer is C. The lac repressor occasionally falls off or debinds, which allows a minimal level of 3 enzymes needed to take up and metabolize lactose, and those 3 enzymes being the beta-galactosidase, the lactose permease, and the transacetylase. So if we go through the other answers here in simple true-false mechanism, if you look at A and B, they mention lactose can freely pass across the cell membrane. We know this is not true. This is through passive transport or facilitated fusion. It cannot pass directly through the cell membrane because the lactose molecule is simply too large. As far as letter D, this is not quite the representation that we usually look for repress the expression of the lac operon. So very simply, there has to be a certain number of the permease proteins always present. Number 3, explain how the lac operon is regulated. Letter A, it is regulated by an activator protein that is activated by allolactose, thus allowing for transcription. Letter B, it is regulated by a repressor protein that is forced to bind by allolactose, thus allowing for transcription. Letter C, it is regulated by an activator protein that must bind to the operator to allow for transcription. Or D, it is regulated by a repressor protein that must be removed from the operator to allow for transcription. Now the answer is both B and D. Very simply, both statements say almost exactly the same thing, which also would lead us to believe that A and C, again, being almost saying the exact same thing, will be false for this type of regulation for the lac operon. So very simply, it is regulated by the repressor protein that is forced to bind by allolactose, so lactose is present. It will allow the transcription to occur. So D is also true, where it's regulated by the repressor protein, and is also must be removed from an operator to allow for transcription to allow the polymerase to begin the reading of the mRNA, to produce the mRNA strand. Activators are A, proteins that must bind to an upstream cis element in order for transcription to be initiated. Letter B, stretches of DNA to which activator proteins must bind in order for transcription to be initiated. C, proteins that bind to the operator region of an operon and allow transcription to be initiated. Or D, proteins that must be removed from an operator in order for transcription to be initiated. The answer is C. Proteins that bind to the operator region of an operon and allow transcription to be initiated are known as activators. Letter A technically is not true because technically the cis element can be a combination of the promoter, the operator, and also the cap binding site. Collectively, they're known as the cis elements for that particular portion of the DNA strand. B is untrue because it is not a stretch of DNA, it actually is a protein. And letter D's definition is given more so for a repressor, not necessarily an activator. 5. Explain why repressors inhibit but activators stimulate transcription. Letter A, repressors bind to specific DNA sequences and inhibit transcription, while activators bind to downstream elements and stimulate transcription. Letter B, repressors bind to activators and remove them from a specific stretch of DNA, which stimulates transcription. Letter C, repressors must be removed from an operator for transcription to proceed, while activators must bind to an operator to stimulate transcription. Or letter D, repressors interact directly with activators, such as allolactose. The answer is C. Repressors must be removed from an operator for transcription to proceed, while activators must bind to an operator to stimulate transcription. So this is a classic example of the LAC operon in use in the early portion of chapter number 7, which talks about the removal of a repressor to allow the RNA polymerase to bind to the promoter and operator in order to actually transcribe the actual mRNA for the enzymes in question. Number 6, the lactose operon exemplifies which central principle of the regulation of gene expression? Letter A, regulation of gene expression is stimulated by repressor proteins and inhibited by activator proteins. Letter B, both positive and negative control of gene expression is mediated by the binding of regulatory proteins to specific DNA sequences. Letter C, regulation of gene expression in prokaryotes is far more complex than regulation of gene expression in eukaryotes. Or letter D, cis-actin control elements guide prokaryotes to allow gene expression to proceed to the termination stage. The only possible answer is letter B. Both positive and negative control of gene expression is mediated by the binding of regulatory proteins to specific DNA sequences, so naturally these are going to be repressors and of course your activators. Letter A is simply a false statement. We know that regulation of gene expression is not stimulated by repressors and therefore also not inhibited by activators. And letter C, regulation of gene expression in prokaryotes is not more complex than for eukaryotes. And while true cis-actin control elements guide prokaryotes to allow gene expression, it does not give us the perfect example of the regulation of gene expression. Number 7, in eukaryotic gene regulation, how do promoters differ from enhancers? Letter A, promoters are regulatory sequences that are usually far upstream from the coding region they control, while enhancers are within a few hundred base pairs of the coding region. B, promoters are often active at low levels, allowing for a minimal level of gene expression, while enhancers are utilized only to upregulate the level of transcription. Letter C, enhancers are strictly on-off regulatory elements, while promoters determine the level of transcription at any given time. Or D, promoters are typically able to bind only specific transcription factors, while a single enhancer may upregulate several different genes. The answer is B, promoters are often active at low levels, allowing for a minimal level of gene expression, while enhancers are utilized only to upregulate the level of transcription. So this is a classic example of one of the figures we had looked at in the second section of chapter 7, which shows the promoter actually allowing transcription of a minimal amount to a particular mRNA strand, whereas an enhancer, whether it be either upstream or downstream or even up to a few kilobases away, can upregulate the level of transcription, and it's not dependent on where exactly the enhancer is in relation to the promoter. As far as the other answers shown here, letter A, again, is false because it does not have to be a few hundred base pairs of the coding region. It can actually be about a few kilobase pairs away from the coding region, so quite far from the initial promoter area. Enhancers themselves are not on-off regulatory elements. Very soon the promoters will do that job. And letter D, the promoters are typically able to bind not only to specific transcription factors, but to general transcription factors. So letter D is false, simply because it is actually a very general transcription factor that a promoter will bind to. Number 8, 3 techniques to isolate transcription factors, which include electrophoretic mobility shift, chromatin precipitation, and DNA affinity chromatography, are based on the same principle. What is that principle, and why would it be effective? Letter A, transcription factors must accurately recognize and bind to a very specific sequence of DNA nucleotides in order to regulate specific gene expression. Letter B, transcription factors are all small molecules, and electrophoretic mobility shift, chromatin precipitation, and DNA affinity chromatography, all separate molecules on the basis of size. Letter C, transcription factors are present in the cell in very small quantities. Abundant proteins would not be detected with any of the techniques. With letter D, transcription factors will bind to multiple promoter regions. All 3 techniques use a variety of promoter sequences at the same time. The answer is A. Transcription factors must accurately recognize and bind to a very specific sequence of DNA nucleotides in order to regulate specific gene expression. So very simply, for all three techniques, we need that specificity in order to isolate a particular type of protein that can be found in each one of the three different techniques. Otherwise, without that specificity, then very simply the accuracy of those particular tests will be shot. Number 9, co-activators act differently than activators and co-repressors act differently than repressors. Which statement correctly contrasts these differences? Letter A, co-activators work together with activators and co-repressors work together with repressors to regulate gene expression. Letter B, co-activators and co-repressors act antagonistically with activators and repressors, respectively, meaning that activity 1 activates or represses the activity of the other. Letter C, co-activators and activators regulate the initiation phase of transcription, while co-repressors and repressors regulate the elongation phase. Letter D, the activators and repressors bind to specific regulatory DNA sequences, while co-activators and co-repressors chemically modify chromosome structure. So letter D most accurately will contrast these particular differences, as the activators and repressors will bind to specific regulatory DNA sequences, while co-activators and co-repressors chemically modify chromosome structure. So very simply, just as it mentions, the activators and repressors will bind directly to the DNA, while the co-activators and co-repressors will basically modify either chromatin structure or they'll interact with mediator as well as other general transcription factors. In the case with co-repressors, it very simply will bind to the mediator and basically prevent it from tracking down the DNA strand and therefore stopping any type of transcription. As far as the other answers here, letter A, while true to a sense, doesn't necessarily give us the entire picture, so it's not the best answer out of the four. Letter B, very simply, the co-activators and co-repressors do not work antagonistically with activators and repressors, they work with them. And letter D, very simply, the second half, which says co-repressors and repressors regulate the elongation phase, that's simply not true. So letter D is the best answer for this question. 10 recent studies have shown that most protein coding genes in the human embryonic stem cells are regulated at the level of elongation by a poised mechanism. How would this be advantageous? Letter A, by poising the RNA polymerase II at the start of elongation, a cell halts all gene expression and has time to develop before activating the genes needed for later steps in differentiation. Letter B, specific internal or external signals would activate a small subset of the poised genes in each cell, thus allowing for specific developmental pathways to be engaged at the proper time. Or letter C, only a small subset of the stem cells in the embryo ever develop. Poisoning allows the embryo to decide which cells will continue to develop and which will not. And letter D, poisoning is necessary to allow for the embryonic stem cells to manufacture necessary biomolecules needed for substantial growth and development. Letter B gives our best answer to this question, as specific internal or external signals would activate a small subset of the poised genes in each cell, thus allowing for the specific developmental pathways to be engaged at the proper time. And the key thing here is when we have that paused transcription of the genes, it is waiting for those internal or external signals to reactivate those particular genes so they can be transcribed. Very simply, the other answers here, both A, C, and D, just don't give us the full picture of how this particular mechanism will be advantageous to the cell. 11, what would happen if eukaryotic transcription were allowed to proceed in the presence of a radio-labeled ATP known as AT32P? Letter A, P-TEFB, positive transcription elongation factor B, will be inactivated by the radioactivity and transcription will be inhibited. Or B, positive transcription elongation factor B will be radio-labeled with 32-phosphate and transcription would not be inhibited. Letter C, multiple factors involved in the elongation phase of transcription will be inactivated by the radioactivity and transcription will be inhibited. Or letter D, multiple factors involved in the elongation phase of transcription will be radio-labeled with 32-phosphate and transcription would not be inhibited. So in this case, the letter D would be your answer, as multiple factors involved in the elongation phase of transcription will be radio-labeled with 32-phosphate and transcription would not be inhibited. So very simply, if you're just simply radio-labeling ATP, it would not inhibit any type of transcription. So we can actually eliminate letter A as well as letter C from possibly being correct. Under letter B, the positive transcription elongation factor B, with a radio-label with the 32-phosphate, the transcription would not be inhibited, but very simply, each mechanism is going to be phosphorylated. In this case, different types of molecules, including NELF, NELF, DSIF, and also the serine of the polymerase CDT, will be actually phosphorylated by the positive transcription elongation factor B. So in this case, letter D is our best answer. 12. Histones are DNA binding proteins with an amino terminal tail that sticks out of the nucleosome. What are the structure and the significance of that exposed tail? Letter A, the amino terminal tail contains a number of amino acids that can be covalently modified by methylation, acetylation, phosphorylation, and ubiquitination. Letter B, the amino terminal tails wrap around the nucleosome and prevent the binding of transcription factors and RNA polymerase II. Letter C, the amino terminal tail contains only structural amino acids. Enzymatic removal of the tail allows for the binding of transcription factors and RNA polymerase II. Or letter D, the amino terminal tail contains a high percentage of hydrophobic amino acid residues, which will recruit activators and RNA polymerase II to active coding regions of the DNA. The answer is A. The amino terminal tail contains a number of amino acids that can be covalently modified by methylation, acetylation, phosphorylation, or ubiquitination. Very simply, that is the sole purpose for these particular amino terminal tails. So very simply, this helps regulate the different areas of either transcriptional activation or repression. The other listed answers here are simply not true. Whereas the amino terminal tails wrap around the nucleosome, that's not true. It's actually free for binding. Letter C, the amino terminal tail contains only structural amino acids. Enzymatic removal of the tail allows for binding transcription factors. That's simply not true. You very well have the different transcription factors binding to that particular amino acid tail. Letter D, the amino terminal tail contains a high percentage of hydrophobic amino acid residues. This is not true. They are mostly hydrophilic, but it does help recruit activators to coding regions of the DNA. Number 13, what is the major difference between histone modification and chromosome remodeling? Letter A, histone modification enzymes excise nucleosomes from the chromatin, while chromosome remodeling enzymes add phosphate, acetyl, or methyl groups to the entire nucleosome. Letter B, the histone modification enzymes remove methyl and acetyl groups from nucleosomes, and chromosome remodeling enzymes replace them. Letter C, histone modification enzymes recruit repressors and activators, while chromosome remodeling enzymes recruit co-repressors and co-activators. Or letter D, histone modification enzymes add side groups to nucleosome proteins, while chromosome remodeling enzymes move, alter, or remove entire nucleosomes. And here the answer is D. Histone modification enzymes add side groups to the nucleosome proteins, again, either activating or repressing certain areas, while chromosome remodeling enzymes move, alter, or remove entire nucleosomes. This is right out of the textbook. It's actually the perfect definition between the histone modification versus the chromosome remodeling. Number 14, what would happen if a cell line were unable to pass on epigenetic changes to progeny cells? Letter A, regulation of gene expression in the progeny cells would not be able to respond to internal or external stimuli, and mitosis would not proceed. Letter B, programs of gene expression developed by the parent cells would not be transmitted to progeny cells during mitosis. Or letter C, the progeny cells would be genetically identical to parent cells and mitosis would proceed normally. Or letter D, the parental cells would be unable to undergo mitosis and the cell line would die, or mitosis would be abnormal and the progeny cells would die. Answer is B. The programs of gene expression developed by the parent cells would not be transmitted to progeny cells during mitosis. So this is the basics of epigenetic inheritance. So very simply, any histone modification from the parent cell gets passed down to the progeny cells with each division of mitosis. So if they were not able to pass down epigenetic changes, it doesn't mean mitosis would not proceed, which means that letter A and letter D simply not true. The mitosis will still occur. Just very simply, if they were not able to pass down the epigenetic changes, it would not be passed down traditionally down to the progeny cells after mitosis. Whereas letter C, progeny cells would be genetically identical to the parent cells. Mitosis would proceed normally. They would not be genetically identical in the sense where the epigenetic inheritance would simply not be transmitted. And finally, number 15, long non-coding RNAs or link RNAs are A, stretches of DNA, aka genes, that do not code RNA, also known as non-coding regions. Letter B, molecules of RNA that bind to DNA coding regions to inhibit the action of histone deacylases, polycomb proteins, and DNA methylases. Or C, molecules of RNA that recruit DNA modifying enzymes and associate them with their binding sites. Or letter D, stretches of DNA that are bound by a variety of histone deacylases, polycomb proteins, and DNA methylases. The answer is C. Molecules of RNA that recruit DNA modifying enzymes and associate them with their binding sites. We can eliminate the other ones here very simply. Letter A, stretches of DNA. We know RNAs are simply not DNA. Very simply, the molecules of RNA that bind DNA coding row genes to inhibit the action of histone deacylases. Glycoproteins, DNA methylases, very simply will not inhibit those particular regions. And we also know, obviously, D stretches DNA. We know RNA and DNA are not the same type of molecule. So letter C is our best answer, as these recruit the DNA modifying enzymes and associate them with their individual bonding sites.